somebody who made a major contribution to science, and there is no exception today. Do you know, John, this will be a question to you, how many people live on our planet?
Hi, John.
John Danielo, how many do you think?
7.2 billion.
I'd say 7.5, but pretty close. But I'd be close.
Very, very close. This is very impressive, John. You're going to get 7 points today, this morning. But the question, which is more important, how many of those people really make a difference and make a change that really impact a very large number of people around the world?
Well, there's 95 of us, 94 of us watching this, so I guess we can be counted.
Yeah, there's a lot of politics with that, Chris. You're venturing into very dangerous territory, Chris. I would be careful.
But there is a one incredible scientist who revolutionized ophthalmology, and we're so lucky we're going to have him presenting his life achievement.
True that. Thanks, Napo.
This is amazing. And with that, let's just move to Anderson.
Hello, everyone. Can everyone hear me okay?
Yes.
Yes.
Okay. Welcome to our weekly ophthalmology research seminar. My name is Anderson Vu. I'm a researcher with Dr. Andrew Brown here at UCI. I have the pleasure today to introduce Dr. Napoleone Ferrara. Dr. Ferrara attained his medical doctorate at the University of Catania Medical School in Catania, Italy, and then began his research career at the University of California, San Francisco's Reproductive Endocrinology Center and Cancer Research Institute. Dr. Ferrara then furthered his research career at Genentech before settling in at UC San Diego. At Genentech, Dr. Ferrara played an integral part in discovering the role of vascular endothelial growth factor in angiogenesis, or VEGF. His groundbreaking discovery in suppressing tumor growth by the inhibition of VEGF with monoclonal antibodies was paramount in the development of bevacizumab, or Avastin, trade name Avastin, as a therapy in various malignancies. This finding also led to research into the role of VEGF in intraocular disease, where Dr. Ferrara also played a vital role in the development of ranibizumab, or Lucentis, a human anti-VEGF antibody fragment. These two therapies are now widely utilized to treat multiple diseases affecting the retina and have fundamentally changed the course of disease, preventing blindness across the globe. Dr. Ferrara is now at UCSD, or UC San Diego School of Medicine, where he holds the position of Hildyard Endowed Chair in Eye Disease, Distinguished Professor of Pathology, Ophthalmology, and Pharmacology, as well as serving as a Senior Deputy Director for Basic Science at the UC San Diego Moores Cancer Center. Dr. Ferrara now investigates alternative mechanisms of tumor and ocular angiogenesis, which we will hear a little bit more about today. With over 200 publications and numerous awards, including being an elected member of the National Academy of Medicine, Breakthrough Prize in Life Sciences, and most recently induction to the Retina Hall of Fame, it is no mystery why we invited him to speak today. Without further ado, please help me welcome Dr. Napoleone Ferrara.
Well, thank you very much for this very, very kind introduction. Let me see if I can get started here. Share screen. Okay. No. Did it work? No. There's some stuff here. Okay. I think we're okay, right? Well, thanks for this great introduction. Actually, really able to do a lot, to live up to this kind of, wonderful words. Actually, so the theme of this. First of all, I want to thank.
Sorry to interrupt, but we can't see your screen. If you're sharing your screen, we can't see it yet.
Oh my goodness. So what did I do wrong? Let me see.
This is always the challenging part, getting that screen sharing going.
Because
sometimes
I think I lost, some, kind of. Okay, let me see. Where was I? Maybe I should try. Oh, okay. I think I'm okay. Yeah. All right. Get all these things out of the way.
There we go. Now it's working.
Wonderful. This is great. Okay, fantastic. Okay, so let me test. Okay, you can
Yes, perfect.
That's great. So I think I want to thank all the organizers, Christoph, Barry, all the all the distinguished people, UCI Ophthalmology. I'm delighted, to speak with you, with such a really wonderful people. Actually, I'm delighted, to meet at least, in the cyberspaces some old, friends like Tony. So the theme of this talk is beyond, VEGF, actually, and I think the truth is that it's been very difficult for anybody who works at least in angiogenesis to go beyond, VEGF, at least in a therapeutic sense. There are still some, but there are still some very interesting and new ideas from multiple groups. I would like to share today some data we very recently published or impressed, in addition to the old, story, all the stuff that probably could be an important background. But I think let me remind you that the neovascularization has been recognized to be as a very important, of course, not only in normal development, in disease. This could be one of the earliest, study that postulate, the existence of an angiogenic factor. A study published in 1939 by a group at University of Rochester. And they used at that time, it was a state-of-art, technology, transparent, chamber. And they implanted tumor so they could, in non-invasive way, observe, the tumor and the vasculature. They observe, as you can see from this precursor of intravitreal microscope, there is an extensive neovascularization which accompanied tumor growth. And this investigator, very insightful, concluded the tumor must be producing some blood vessel growth, stimulating factor. Actually, this was a very innovative idea. And then just a few years later, in 1948, Dr. Michelson made this seminal observation, which of course is very well known to everyone who is interested in the retina, in the eye. The idea that there is a factor X, produced by the retina. Now we know it's induced by the ischemic retina, and this was a very, very critical idea, extremely important idea in the field, which drove, the field. Many decades later, we have actually much better idea how angiogenesis regulate. There is actually, we know there is work done in many labs, over the last, show that there is like a series of key cellular and molecular play that regulate angiogenesis. It looks like, this slides illustrate, some of those. Look like an orchestra. Everything needs to be perfectly timed, to have a normal vessel wall, to have the differentiation in arteries and in veins, and as well as in investment of endothelial cells by mural cells like pericytes. So this is clearly play a critical role. And there is a many player, not only by no means, VEGF. There is like the heparins, notch, PDGF, etc., etc. So it's a very complex process. But in terms of a therapeutic target, that would be probably VEGF, that would be an important molecule, becoming an especially important molecule. This is just, highlight. I stumbled into this field, when I was working in reproductive endocrinology at UCSD, and I was able to identify some cells called follicular cells, pituitary follicular cells. Still very mysterious cells. But I observed that they release a mitogenic factor for endothelial cells, and then I was young and irresponsible, so I want to pursue isolating a protein, even though I never lots of back training. But I learned on the job, and then when I joined Genentech, we were able to actually to finish this work, by determining the N-terminal sequence and the cDNA of this molecule, which is now called VEGF. Of course, we need to acknowledge other investigators like Dr. Hal Dvorak. We had independent isolating of VPF. At the time, they couldn't isolate, the protein for fully, so it did not have any identity as a molecule. But after this work was published, it became clear that VEGF and VPF were the same molecule. There are some interesting features about VEGF, which is first sort of provide a sort of prototype, how angiogenesis regulate. As you can see, there is the VEGF gene can undergoes alternative splicing, which is nothing uncommon in that. But this is generated different isoform, which have a different, very significant by one property, which is affinity for heparin. It goes from a very little or no affinity in VEGF 121 to have the highest affinity in VEGF 206, and which is a 2 heparin binding domain. And probably the most important member of this family actually is a VEGF 165, which seems to have is only 1 heparin binding domain. So seems to have achieved a perfect balance actually of a diffusibility and binding to the extracellular matrix, which is critical to build gradient. And another event which is important is proteolysis. But what I would like to emphasize how the interaction with the matrix with the heparan sulfate proteoglycan is so critical to build this angiogenic gradient, this tridimensional gradient, which is so essential for a correct formation of the vessel wall. Of all the experiment that you could do to emphasize the importance of VEGF in angiogenesis, I don't think I cannot do really any better than this, even though it was published 25 years ago. At the same, and the Peter Carmelit group also reported the same similar finding in the same issue of Nature. Actually, we show there is this striking heterozygous lethality. If you inactivate only one VEGF allele, there is this defective vascularization in the yolk sac, and an embryonic lethality at day 10.5, which is very highly unusual, at least in the mammalian genome. Another interesting aspect, is about the VEGF isoform. Actually, let me go back to them. Actually, there is some work which is raised the possibility you not only have the pro-angiogenic isoform, but there is actually anti-angiogenic isoform. By this work by David Bates, which generated lots of debate. He claimed that there was a 165B, but which inhibit angiogenesis. And recently there is some publication that kind of totally refute the existence of this molecule. Another story which I thought was very intriguing, actually, piqued my curiosity even more than that. There is a publication by Paul Fox and colleague at the Cleveland Clinic. They report by very different mechanisms, by programmed read-through translation. There is the generation of an extended form of VEGF, which is also anti-angiogenic. By read-through translation, a stop codon can be read, as a sense, code, and there is a serine, and is read as a serine. This extension, which they report, to be anti-angiogenic. Actually, we spent a little bit of time because at the time it was really intriguing. Actually, I want, mostly I want, to train and to make sure that people in our lab at UC San Diego became more familiar with some biochemistry, some assay. And we're able to show that this molecule, if it truly exists, actually does not have any anti-angiogenic characteristic. Actually, it's probably a weaker agonist because it does not bind with the neuropilin. So this is another really myth about this anti-angiogenic form which has been dispelled. So this is kind of interesting. So far, maybe boring as it may sound, VEGF is only pro-angiogenic. And this is actually illustrating the VEGF path at a review that I have very much the pleasure to co-author with Tony a few years ago. Actually, we summarized about the basic biology and oncology and ophthalmology. And without going into many detail, because these things I think are very well known, we know that there is, in addition to VEGF, which is called now VEGFA, you have a variety of other some VEGF-related gene, like VEGF, B and PLGF, as well as VEGF-C and D. Interesting, VEGF interacted with the two receptors, VEGF receptor 2 and VEGF receptor 1. A VEGF receptor 2 now is thought to be the key signaling receptor, which mediates all of the activity of VEGF, like angiogenesis, mitogenesis, and permeability. And VEGF receptor 1 is still a very enigmatic molecule physiologically because it interacts selectively with the VEGF-B and PLGF. One feature of VEGF, actually, we're very fortunate to discover that in the early 90s in collaboration with Rusty Williams. We identified this FIMS-like tyrosine kinase receptor for VEGF. At the time, it was striking. It did not really lead to VEGF, dependent tyrosine phosphorylase. We almost missed that. Instead, this molecule binds VEGF like a gangbuster, but there is with a very high affinity, very high capacity. And actually, we show that in a study, the domain 2 is the key binding element. And this is the study. Actually, I must say, was the foundation of the work done by Regeneron that that actually develop a soluble receptor, those as aflibercept, based on this binding of domain 2 to VEGF. But clearly, there is a lot of complexity. There are also some co-receptor, and interesting, there is also another receptor, which is VEGF receptor 3, which is regulating lymph-angiogenesis. So it's very striking that the same, pathway, the same, tyrosine kinase, the VEGF receptor mediated both, hemangiogenesis and lymph-angiogenesis. But clearly, in the major interest, in this pathway has been as a therapeutic target. Of course, there is lots of interest in basic biology. This could be one of the earliest, study, maybe perhaps, the first study to prove that blocking of VEGF, mediate angiogenesis a benefit, in cancer patients. Actually, this was the study was presented at ASCO in 2003, and it was published in 2004. This was the time when there was lots of skepticism about the anti-angiogenic approach. There has been a number of, unsuccessful clinical trials. The study showed that actually, if you block, VEGF with an antibody, with a humanized antibody called Avastin, which I'm sure many people are well aware of this drug because it's been widely used also in ophthalmology. There was a survival benefit, in patients, actually. And actually, years later, now we know that bevacizumab or Avastin, which was first approved in 2004, for colorectal cancer, is widely used, as a cancer therapy. And not only that, it's widely used also in ophthalmology. Is off-label use, is today is a 12 FDA approval in different indication in multiple tumor types. Actually, in 2018, bevacizumab was one of the top among the top 10 drugs for sales. And I don't know if it's still true today because there are some biosimilar which has been approved. But certainly remain one of the most widely used therapeutics, which is the fact that there is some biosimilar. I'm sorry to say this to Tony, who still works at Genentech. I think it's great to provide a new life that it makes this drug still available to patients. And clearly there was a lot of interest, of course, to extend, the efficacy of bevacizumab, in cancer and potentially in other diseases, actually by combination. One of the most, rational combination would have been like combining with the vascular targets. There is a number of targets in the vessel wall, like the Tie2, angiopoietins, as well as the PDGF, and as well as the integrins, like alpha V beta 3, alpha V beta 1. And there is a lot of nice preclinical data which suggests that. What one can say that really, unfortunately, we all disappointed with that, because if any of the study had been successful, probably this would have advanced the field. Unfortunately, it has been very difficult to demonstrate any additivity, with the target. For example, a few years ago, you could have not gone to any cancer meeting or even without hearing what a fantastic target was CMET, and yet all the clinical trial with the CMET fail, including a combination with anti-VEGF, in colorectal cancer, in lung cancer, was no benefit. Even angiopoietin 2, a bispecific and monoclonal antibody with targeted VEGF and angiopoietin 2, at least in cancer, it didn't result in any benefit. Without going to any, this has been true for all of these targets. So it was a little bit, bleak that it was impossible to advance, the therapy with a combination with this seems to be very rational. What has been, however, very interesting, that has been actually in the year of immunotherapy, actually, it's been possible to find some very important, very clinical, meaningful additivity. Even though this is a group works in ophthalmology, I believe it's very well known that in the immunocheckpoint inhibitor like anti-PD-1, anti-PD-1, and anti-CTLA4, clearly we say the measure advance in cancer therapy. And actually, there has been actually interest in testing whether they will be combined with anti-angiogenic therapy. This is a slide actually from one of the Ira Mellman's review that actually illustrated the many possible ways how VEGF actually could synergize with immunotherapy. Most of these things actually, the fact that there's so many possibilities show that probably very few are true. But clearly, people have shown very consistently, if you block VEGF, there is an increase in tumor infiltrate lymphocyte in the tumor, which maybe provide a substrate for this additivity. activity. And there are already 2 FDA approvals of bevacizumab with anti-PD-L1 antibody. One was happened in 2018 in non-small cell lung cancer. I want only to show the most recent one, which is in hepatocellular carcinoma, which is a tumor which is has been very difficult to treat, especially in very widespread, for example, in Asia. For the last, more than a decade, there has been absolutely no advance in therapy. The standard of therapy was sorafenib, which is actually a significant, but not dramatic survival benefit. And the combination of Avastin and Tecentriq, which is the name of this drug, resulted in nice survival benefit, comparing to sorafenib. So this is very, very nice. Actually, there has been this additivity. However, our lab has been interested not so much in adaptive immune system, but in the innate immune system. And this work, while it was focused on cancer, it could have certainly application even in other, even in eye disorders, because you can find many of the same player in the retina. Actually, at the time, actually, we're interested, in identify some tumor cell line which are either not very sensitive or moderately sensitive to anti-VEGF. At the time, this was made possible by some phage-derived antibody which block, VEGF across, species, unlike bevacizumab, which is species-specific. We identify this. You can see this cell line in yellow, which is only maybe 30% inhibition with this anti-VEGF antibody, and 2 cell line, which is much greater level of responsiveness, which is the TAB6 and B16F1. And actually, this is where our model, we're hoping that by comparing and contrasting the cell line, we could learn something which could be actually interesting, And actually, we apply a model which has been widely used, which is a GFP transgenic, making transplanting bone marrow from GFP transgenic mice, into a lethally irradiated host, So it's got to be everything which comes to the bone marrow is green. Actually, we published the study a few years ago, and we found a kind of striking that the sensitive tumor have very few green elements, which means bone marrow derived element in the tumor microenvironment. In contrast, to the resistant tumor, you have lots of green elements actually, and most of these cells actually belong to a class, CD11b GR1 myeloid cells, and which is a broad spectrum of cells, which include mostly cells of the neutrophil lineage, also monocyte and dendritic cells, but predominantly they are neutrophils. Actually, this slides illustrate actually the subset of CD11b GR1 positive cells based on this mark. You have Ly6C positive cells, which are mostly macrophage and dendritic cells, and Ly6G, which are mostly, as I said, of the neutrophil lineage. There is actually a subset of cells which are called myeloid-derived suppressor cells, which actually are being reported to inhibit, T-cell mediated function. Actually, and actually, to make a long story short, we found that this immunosuppression didn't play really any role, because we saw exactly the same effect, in resistance or sensitivity to anti-VEGF, both in syngeneic mice and in the skid mice. We're able to identify actually a key molecular pathway. That's I hope it goes to the concept of beyond VEGF, produced by these neutrophils, by these activated neutrophils. It's a molecule actually I identified years ago, actually based on a genomic screen. Actually, we found these 2 peptides, we call EGVEGF and BV8, which is prokinase 1 and 2. They are 2 small peptides. Actually, this is the structure of a colipase fold. Colipase is an enzyme, even though this molecule don't have this enzyme activity. They have a motif in the terminus, this AVIT, which seems to be critical for biological activity and defy this family. These two molecules, EGVEGF and BV8, or aka PKR1 and PKR2, actually highly conserved, and actually interacted with the 2 highly related GPCRs, called PKR1 and PKR2. Actually, this system is already a rolling disease. They've been characterized as a neuropeptide. They've been implicated in circadian rhythm, olfactory bulb neurogenesis, and in some cases even in Kalman's syndrome, which is a disease actually in olfactory bulb neurogenesis, and the generation neurons have a similar origin. So there is a combination actually of olfactory bulb defect, and infertility, which is characterized Kalman's syndrome. And actually, years ago, we were able to show that blocking this BV8 with an antibody reduce actually angiogenesis, actually not maybe to the same extent as anti-VEGF, in a very significant way. And actually, we followed the story actually very doggedly for many years. It started in 2017. We published a paper very recently. I will very briefly describe it. Actually, it was published a week ago. I thought it was a very interesting biology actually, and one of the limitations of this approach actually, there was a technical limitation that prevented from going to the clinic. It was very difficult to make a single antibody that block BV8. Actually, you need always to combine two antibodies to have a robust effect. This is by no means unprecedented. For example, hepatocyte growth factor is another molecule which is very difficult to block, but make it very difficult actually to go to the clinic and be a deal breaker if you want to go to the clinic having two antibodies. But actually, we found, actually, we discovered there is the RAS pathway plays a critical role, in regulating the system. We found, there is the key myeloid growth factor, GCSF, is critically implicated in that. GCSF actually mobilize and expand the myeloid, progenitors of the neutrophil lineage, a strong upregulating of BV8 and block, this molecule actually inhibits angiogenesis. Actually, we're very intriguing, because most of the study was done in xenografts, which have a number of theoretical limitations. Work we did actually at UCSF, actually in collaboration with Dr. Taketo in Kyoto University, and a couple of visiting scientists from Japan. We're interested to see what would be the mechanism of resistance to anti-VEGF in a very complex genetic model of colon cancer, which mutation which recapitulate this mutation like APC, SMAD4 knockout, and activation of a KRAS. This is kind of the classical Vogelgram. And actually, we're able to show that actually in VEGF inhibition plays a role. Actually, in inhibition of this tumor growth. Even with the KRAS activated with SMAD4 knockout. Actually, so in spite of this complexity of this genetic complex, still anti-VEGF has an effect in this model, which is not exactly what you see in the clinic when you need to combine it with the chemotherapy. And we thought that probably this model missed some key component, which is true in tumors. It could be inflammation actually, and actually we use a model actually which has been utilized by many investigators, giving dextran sulfate, which actually strongly promoting inflammation, mostly in the colon. And what is very remarkable in all this model with the APC mean mutation, actually, even all the tumors are in the small intestine, not in the colon, which is really very unusual, very different, which is clearly a difference for the human disease. But instead, if you give DSS, you give this dextran sulfate, it promote inflammation. You have the appearance of this tumors in the colon, which actually, and we thought that this could be potentially very interesting model. And we're able to demonstrate actually that when you have a colon inflammation, actually, these tumors now become resistant to anti-VEGF. Actually, this is what is very, very intriguing that tumors in the small intestine, actually, where there is a much less inflammation, even with this DSS, remain a strongly response to anti-VEGF. So there is something very unique about this pro-inflammatory milieu. Actually, this is very busy slide. I'm not going to go in in enormous detail. We found, re-encounter, even though we're not, looking for that. Our old, friends, GCSF and BVA. There is a very strong increase in the serum level of GCSF, in the tumor-bearing mice. Actually, we've been treated with the DSS, And at this point, the combination of an anti-VEGF, with either GCSF or BVA is a very remarkable inhibitory effect as compared to the control. And actually, we've been able to show we're able to go even a step further. Actually, most of this tumor recapitulated primary tumor growth, but there is no metastasis. So with our colleague in our lab, we took advantage of a model actually of a metastasis model, which include a very complex genetic model with the Villing Creek activating KRAS and P10 knockout. In this case, there is metastasis, even though very infrequent, take a very long time to develop. And we're able to show that in this metastasis, there is strong upregulation of a GCSF, and there is lots of tumor infiltrating neutrophils actually with expressing BV8. We didn't attempt, to do a blocking experiment because it would take another year or longer because of given the low frequency of metastasis in this model. But clearly, it's very intriguing. And actually, work we did when I was still at Genentech, actually showed that this system there is a very strong correlate to the human disease. For example, there is a tumor infiltrated neutrophils in colon cancer strongly express BV8. This is a score show that the majority of case there is of this using the tissue microarray there is expression of BV8. And actually, interestingly, this IBV8 is a prognostic factor for survival in patients with the colon cancer. We're able to measure the level of this protein in the plasma of patient from the original colorectal cancer, in the Urwitz et al. This is a negative survival prognostic factor. Higher BV8, actually predict, reducing survival. And so, this is slide illustrates how myeloid cells, and in particular in neutrophils, can affect tumor growth. Of course, this is not limited to tumor growth. They produce VEGF, also produce a series of mediators which actually can also directly stimulate mitogenesis and angiogenesis. So this actually, to summarize this part of this work, resistant to anti-VEGF is not associated with the mutation, but this pro-inflammatory cells, myeloid cells, can actually play a very critical role. We identify a mediator of the cells, a key mediator of these cells, which maybe one day will be a therapeutic target if we can find a good inhibitor and go to the clinic. Of course, naturally, probably the application of anti-VEGF, which has the greatest impact, has been probably has been the eye. Actually, as we all know, there is a robust effect in cancer, but the greatest benefit has been in the eye. I don't need to this audience to emphasize the difference between ischemic retinal disease and AMD actually, and wet AMD. AMD actually, wet AMD has been historically an intractable disease actually, but work done by multiple group actually. I want to acknowledge, of course, the work by Tony actually was a pioneer, started with the mitogen and as well as others. It's been possible to show that the anti-VEGF has really unexpected impact in AMD, much greater than probably would have expected, much greater than in cancer actually. In the study actually, in the anchor study actually, in a very aggressive form of AMD actually, the classical form of AMD, blocking of VEGF with the ranibizumab, is resulting in increase in visual acuity in about 35% of patients, which drives these curves. And this was the standard of therapy, actually, the photodynamic therapy. And the study has been actually confirmed. There is a multiple study with a multiple phase 3 study, which have validated the benefit of anti-VEGF. Actually, this illustrated some concept actually of of vascular biology show that blocking of VEGF not only has a profound inhibitory effect on expanded the choroidal neovascularization greater than cancer. This in this setting there is probably less room for escape. You don't have the genomic instability. But what is very remarkable is the fact that you can prune these vessels which are structurally abnormal. They lack pericyte. They tend to leak and bleed. And they create a vasculature which is less prone to bleeding. We have also, as many people know, some long-term data which illustrate the benefit of anti-VEGF. This is the 5-year CAT study with comparing ranibizumab and bevacizumab. Show that about 50% of patients a good visual acuity, 10% of 20/20 vision. And this data should be certainly put in the context of what was known before, which was the therapy before anti-VEGF agents were available. Actually, at that time, it would have been very rare to find anybody with a good vision. So this is very, I believe, a significant advance in the field. And there is a population data which actually confirmed this data. Actually, this study published a few years ago illustrated the incident rate of legal blindness due to either to AMD or to other cause, other disease, in this time frame. And they showed that after 2006, when the anti-VEGF therapy became available, there has been a 50% drop in the incidence rate of legal blindness, which actually it has been seen in other cases. Actually, it showed this the impact of anti-VEGF. And actually, in the attempt to go beyond VEGF, actually clearly that not all patients responded to anti-VEGF therapy. There will be certainly need to do even better. And there will be some really very commendable effort to do that. For example, we combine anti-VEGF with the anti-PDGF. There is a rationale for doing that. That you can prune the vessels which are pericyte poor. You can regress the vasculature. And there was some promising data, but unfortunately this approach did not work. And this data keep always in our thoughts. It illustrated the complexity of biology, even though there is some intriguing preclinical data. It's not easy to go beyond that. Another really interesting area of biology is the combination of anti-VEGF actually with the Tie2 with the Tie2 pathway. This approach has not been dramatically successful in cancer, but there is actually some hope. That maybe in ophthalmology can result in some benefit. There will be some mixed data combining, for example, in aflibercept with an antibody which will target PDGFB, and didn't result in any benefit. But Genentech as a bispecific antibody which is slightly different from the one used in cancer. And there are some intriguing data, some promising data which show that there is perhaps some benefit, but time will tell the extent of this benefit. Clearly, there is some clearly genetic, the AMD is as everyone knows, is a spectrum of disease, ranging from early to advanced AMD. And there is a very strong, genetic component. There is some key risk, factor. For example, one is complement factor H, and like HTR1 actually. This can result in a very dramatic increase in risk, several-fold increase in AMD for patients with this mutation. And that's led, to some number of clinical trials actually still in course. One was actually with the lampalizumab, which inhibits, the complement, pathway and the anti-factor D antibody. And once again, in spite of all the hopes, and this very intriguing phase 2 data, we can say that, unfortunately, there has been no benefit, as of today. So there is still an unmet need, to find a better way to treat AMD. And actually, in the last few minutes, I would like to show you some data. Actually, still unpublished. Actually, are impressed. Actually, I don't know yet the clinical relevance, but suggest that there is still the possibility you find at least at the level of pure research you find additional mediator, additional molecule. this is an observation we made a number of years ago. Actually, we found actually that in addition to the protein growth factor like a VEGF, basic FGF, there could be some molecule of a totally different nature, different class. We found that a number of tumor cell line produce an angiogenic factor. So far, of course, there is nothing surprising to that. There is nothing unusual into that. What was very surprising that this mitogenic activity went through 3K filters, 1K filter, even 500 kilodantal filter. So it was a very small molecule, which didn't seem to fit in any known class. Actually, this molecule actually was a very hydrophilic molecule. They didn't bind to the column like a C18. Actually, showed that this molecule happened to be, there could be extremely challenged to isolate. We spent a considerable amount of time, trying to purify this molecule. That would be very maybe overly ambitious project. There was a number of technical challenge, like the fact that the interference with the endothelial cell proliferation assay with of the buffer used for this chromatography. And so and unless we gave a better idea of how to do that, we put this project on hold. But we said if we cannot do plan A, maybe we might as well do plan B. And actually, we thought that it could be that a screening library of a small molecule, we can find some molecules which are interesting biological activity. Actually, we found a library actually of a small molecule. They are mostly mass spec standard, high quality library. Now it's been marketed by Sigma, initially from a smaller company. And then we thought of testing this molecule for the ability to stimulate growth of choroidal endothelial cells, actually bovine choroidal endothelial cells. Endothelial cells are always in our mind, actually, because this could be particularly interesting. And we found actually an interesting molecule, totally unexpectedly. That's what you find really when you do this kind of screen, you can expect it unexpected. There is a molecule called mannosamine, which is a variant of mannose with the amino group. It actually could be an intermediate in the center of a sialic acid. Actually, this illustrates the tridimensional organization and the structure of mannosamine. It is molecule, to our surprise, stimulate endothelial cell growth. Maybe the effect was mostly notable in combination with the VEGF. Actually, this illustrates the effect. No VEGF, it is VEGF alone, and this the combination with the mannosamine. It is very clearly an additivity. This is seen in multiple model, actually, in multiple setting. And instead, other related exosamine completely lack, this effect. For example, this glucosamine, mannose, etc., and others seems to be very unique, to form mannosamine. And you certainly, actually, this is another assay. This is a scratch assay, which show that actually, once again, the combination of mannosamine and VEGF, crosses the gap, more rapidly. And actually, there is a lot of data here, so we're almost running out of time. So just say that the paper should be coming out very soon. We found that actually mannosamine selective activated the JNK pathway in endothelial cells. Actually, we found that it activated the UPR, the unfolded protein response, and stimulate the JNK pathway. JNK inhibitor inhibit this effect of mannosamine. Actually, mannosamine unlike mannosamine, it does not activate the JNK pathway. And this could be a unique aspect of this molecule. Actually, it is illustrated kind of a diagram. It is cartoon illustrated some potential actually mechanism of action. Actually, there is an inhibition of a lipid linked, oligosaccharide, which this is in fact been already shown for mannosamine, and it inhibits glycosylation and then activate, by inducing BIP, activate, the resulting in mild ER stress, which stimulate angiogenesis. And actually, we're able to show that in complex in vivo system, this is relevant. This is, for example, we showed that topic administration, the mannose-amine and VEGF, promoted wound healing in a mouse model. Interesting, also stimulate actually blood flow recovery in the mouse model of ischemic limb. Actually, this is kind of we tested this model because in the study conducted in the 90s, actually, which unfortunately were not very successful in the clinical setting, showed that VEGF stimulate in limb ischemia, collateral growth in limb ischemia model. But what we found very intriguing is that actually mannose-amine also when injected intravitreally stimulate retinal angiogenesis in mice. Actually, it was a very significant effect. Almost similar to comparable to basic FGF. And actually, what is interesting that another glycosylation inhibitor like KIF, which is a very similar effect as mannose-amine, also stimulate angiogenesis. Actually, and actually we don't know what is the relevance, but we think that perhaps it's worth to explore this effect, because one of the things that happen in geographic atrophy in dry AMD is the drop of a choriocapillaris. So perhaps this at this point is totally speculative, but actually would be interesting to test whether it was this kind of approach, which I would say, unlike VEGF, does not induce permeability, does not induce any vascular leakage, might have perhaps a benefit in this condition. But this at this point is a pure basic research, in the spirit of actually going anti-VEGF, presenting new ideas actually. So I will conclude this, say that VEGF has had a very significant impact in disease. There is a significant benefit in cancer, in eye disease. But one of the challenges been actually, among other things, find optimal treatment and in combination. We found a combination with immunotherapy in cancer, and in the eye it's been so far very difficult to find this additivity with anti-VEGF. But clearly there is actually other avenues that other studying other pathways which provide other targets, perhaps potentially using this molecule, like for example a molecule like a mannose-amine. Probably there are others. One could have a benefit in a variety of conditions. And with this, I would like to conclude this talk. Actually acknowledge many colleagues in collaborator. We actually this actually the more recent colleague at UCSF. I'm also including Pretty Egde, Frank Pilla, Jen and Tek that did this work on BV8 in colon cancer, and this work is going on for decades. So it would be impossible to acknowledge everybody. And I would like to thank you very much for your attention. I'm very happy to answer any question you may have.
Thank you very much. If there are any questions, please drop me a chat note and we go as they will come. So anybody? Let's give a minute until the thoughts are getting together.
No problem at all.
Okay, Daniel.
Yes, hi Napo, this is Dan Chow from UCSD. Really enjoyed your overview.
Hey Dan, how are you?
Good to see you. Good to see you. So I had a broad question. So there's a strong disconnect between very strong preclinical data showing additive pathways to VEGF and the lack of response in clinical trials. And I was wondering if you could
You're talking about the cancer now, right? Or
cancer, or let's say eye disease, let's say like PDGF or any of these things. And I wonder if you could speculate on potentially why that might be. And one thought I had is that are there any human-specific stimulators or inhibitors of angiogenesis that are not present, for instance, in the mouse, that could be targeted?
Well, that is certainly conceivable. Actually, yeah. First of all, I would like to say there is in the human there is a complex. For example, the targeting pericytes. Even though it was a very promising approach, but seems to be like a double-edged sword, because if you block pericyte, actually studying cancer show that on one hand you regress the blood vessels, you can make a smaller tumor. At the same time, you can promote metastasis, because if you create this imperfect, dysfunction of microvasculature, etc. You could make the same case in the eye, etc. But in the idea that you can have a mediator which are not, it is totally possible. For example, there are molecules, for example, like for example, allele, it does not exist in mice. There is some chemokine, especially chemokine and cytokine, this species specificity. Actually, we try to verify everything that we did, especially for this BV8, because we're very concerned that could be the case. But instead of the system, at least in a descriptive way, in a correlative way, seems to be well conserved. But yes, there is a number of pitfalls in trying to translate mouse to humans. There is a lot of possibilities, I guess.
So I have a quick question to you. You mentioned about this isoform of VEGF, multiple of them, and you touch on that a little bit in your presentation. What do you think? Why evolutionarily so many were formed? It's not characteristic to every gene. It's just to VEGF is so many.
Well, I think that there is you're talking about the classical. I think it turned out to be that's very. They seem to mediate different function actually. Unlike, for example, genes in which encode protein which is mostly an intracellular function or cell-associated function. VEGF is a molecule mostly operate in the extracellular milieu. It's a very critical actually aspect of that. So the alternative splice, what it does mostly is a generative kind of a gradient of heparin affinity, which really regulate and modulate the bioavailability, the diffusibility of this molecule. And now we know that over this isoform of VEGF 165, which is one heparin binding domain, is absolutely essential for the development of vasculature, etc. There is a genetic data to show that if you have only a mutant which expresses only VEGF 165 is normal, because once again it's very critical actually, is a diffuse, it created this biochemical gradient, but at the same time it is not so tightly associated, which actually it does not work. To get to your point, why do we have VEGF 189? It's not really totally clear why actually I don't know, but because it seems that VEGF 165 is the only one which is truly essential. Maybe there is some function could be storing an angiogenic, this angiogenic molecule which is bound to the ECM could be bioavailable when there is a proteolytic event. This could be the only thing that I can think about. But the most compelling evidence is for VEGF 165.
Mm-hmm. Dorota, do you have any question?
Yes, actually I do. I was wondering about the potential usability of new molecules, and especially the one that you described in the ischemic stress and stroke, maybe in retina first, but maybe then in brain to rebuild the vasculature, especially the small capillary.
I don't know, that's a great idea. We don't really, we've been working for the last, maybe a couple of years on this molecule, so this only, you cannot answer all the questions, probably will take years. But this is all of it to us a very intriguing possibility of a molecule that actually by novel mechanism, like this mild ER express, you can actually promoting survival and growth of endothelium. If you go to very high concentration, then you have the opposite effect. There is a sweating spot in the dose response. But conceivably, we're going to test that. We try to apply for grants. So that's one of the things that when I was at Genentech, I didn't have to do that, but now we have to do that. And so we try to actually come up with some of these models. Actually, we found that this model, for example, among other things, is very stable to bacterial environment. One of the reviewers asking this question for wound healing, so in resistance to it in a model with the Staphylococcus aureus. So it might play a role actually in the treatment of infecting wounds. It probably a molecule like a VEGF was not successful because it probably degraded and inactivated. So there is a number of possibilities. I can certainly the retina and the central nervous system could be targeted with this molecule. So I'm very excited to test these possibilities.
Fantastic.
Thank you.
We have two other questions. Eric first, and my favorite, Corinne. Eric?
Yeah, this is Eric Perlman. I really enjoyed the talk. and especially interested in the glycosylation aspect of this, or at least the Mann oscillation side of things, as you're discussing this now. But wondering if knowing that this changes the amount of carbohydrates on our cells, and the glycocalyx changes as we age. I think, if I remember correctly, it's reduced. Do you think this might have a role in AMD?
Well, I don't know really. We're very interested in exploring that. I don't know if it by itself is a role in it, play a role in it, because this is kind of a tool. Kind of this molecule probably is not clear that even exists in physiology, or it's very quickly formed intermediate. Was initially described in the cell wall of bacteria. Probably is a very transient intermediate in mammalian cells. But clearly, I'll view this mostly as a pharmacological tool that you could perhaps create an ER space. And then maybe actually my favorite idea, I don't know if it turned out to be right or wrong, is you could with this tool protect the choriocapillaris. There is a lots of study actually that implicate like a dropout in the choriocapillaris, at least one of the events in the geographic atrophy, and maybe perhaps maybe that a molecule like this could have a role. You could not use VEGF in that, I believe, because that could have the effect of permeability, acute effect, etc. And we'll try to test this possibility.
You showed the same effect with kifenensin, which has a broader role in blocking carbohydrate formation.
Yeah, exactly. This inhibitor, which also could cause ER space, etc. They all actually they all do the same thing. They can do that, because so we believe that it's a broader mechanism, but not limited to mannose. To this glycosylation inhibitor, they all seems to be able to induce ER space.
All right, Corinne.
Actually, thank you for the overview. It was very exciting and very additional knowledge. But my question goes back to minosamine, and you might have answered it already. But let's just confirm, you have not tested this in any of the common models of ocular diseases, have you yet?
No, we only not in the ocular. We only test. As far as that is concerned, actually, we did an intravitreal injection of mannose. It was a robust effect, a very robust effect. As far as disease model, we only test, in the limb ischemia model as well as in the wound healing model. But we have because this model we're easily available to us, etc. So, but we are certainly eager to test in this eye disease model. Unfortunately, there are not that many actually. Maybe perhaps the sodium iodate model. That's one of the things that we thought. If you have any advice, if any suggestion, would be great. Whether it's this kind of this toxic effect, this antioxidant effect, which destroy the vasculature at RPE. So we'll see if there's any beneficial effect, any pro-survival effect.
Well, there's probably more appropriate models like the CNV models. Probably might be more appropriate than the sodium iodate model.
The CMV model is a model already. There is such a strong neovascularization. What we thought the application of this model is not to inhibit angiogenesis, but if anything to stimulate, to promote an endothelial survival. In the CMV model, this enormous hypoxic stimulus of VEGF. So maybe that in models where actually the vessels are kind of smashed out, they're destroyed by this abnormal microenvironment and maybe protected. But this at this point is total speculation.
So at this time, we need to find a blocker of minosamine, actually.
Well, I don't know, because minosamine it is not there, because of the, probably I think minosamine could be a tool actually to protect blood vessels as opposed to to try to block it.
Mm-hmm. Thanks,
John Hong. Hello, John.
Looks like Dorota has another question.
Yeah, I just wanted to continue upon Corinne's note and about the eye diseases. Actually, in aging, the vessels are extremely fragile, and I was wondering if you thought about potential application in aged animals or any other model that would help maybe survive such a problems as exactly the diabetic retinopathy, or maybe ischemic stress, glaucoma-related vessel break. So that's actually very exciting for other types of diseases, and maybe we can discuss about it. I would be very interested maybe to try it.
Great idea, great suggestion actually. And they're all testable hypotheses, because a manosamine can be orally administered. Actually, we give it orally in actually. Now in the wound healing study we give locally, but like in the limb ischemia model we give it actually orally, like a sugar. You can actually administer the same PK as other sugar. So it's very easy to do that. So this all great idea actually. One of the things that we have proposed in a grant application to test in a variety of conditions where you kind of want to protect the blood vessels, endothelial cells when they're fragile, etc. So, but your point's absolutely well taken. Actually, if you have any model in mind, I'll be happy to talk with you more. Maybe after that, we can just connect, etc.
Absolutely, I actually think we could be great.
Yeah.
All right, thank you very much. We have one eager person who is Barry Cooperman, and he will give a final words.
Okay, well, thank you, Napo, for that great presentation. There's a million more questions to ask, but again, your overview was fantastic. Still want to know more about the inflammatory aspect of diabetic macular edema and why VEGF doesn't affect those other cytokines, but there's so much contribution you've done. Thank you for all you've done for our field, and look forward to our ongoing discussions. Appreciate your time and enthusiasm so much.
It's been delighted to talk to you today. It's been fantastic. I really look forward, even to connect more and discuss. Hopefully we can lead to some collaboration, etc.
We'd love to do that. All right. Thank you, everyone. Thank you, Napo. Appreciate it. Thank you, Chris, for organizing the series as always.